So today I'll be talking about the regulation of genetically engineered microbes at the Environmental Protection Agency. As they introduced me, I am Kara Welch, and I work in the Emerging Technologies branch in the Office of Pesticide Programs at the Environmental Protection Agency. A mouthful, but for anyone looking to register a genetically engineered microbial pesticide, the Emerging Technologies branch would be the first step for discussions with EPA. We're responsible for the registration and risk assessment of everything from GE microbes, our focus today, modified mosquito products, plant incorporated protectants, and other genetically modified or emergent technologies. But today I'll be going over what types of GE microbial products are under the authority of our group, and in addition, what the pesticide registration process looks like. So if we move to the next slide. As we move through, I'm going to start with the basics and then follow up with the regulatory pathways for GE microbes in particular. We'll go over quickly what a pesticidal active ingredient is, what statutes our branch and the EPA oversees pesticides through, and then specifics of data requirements and best practices. Next slide. This may be something that many of the individuals here are aware of, but I wanted to start with defining a pesticidal active ingredient as it's stated in FIFRA, the Federal Insecticide, Fungicide, and Rodenticide Act. You can see it here on the slide, bolded and underlined. It's any substance that will prevent, destroy, or repel, or mitigate any pest. EPA oversees both microbial pesticides as well as GEMS, GE microbes. Largely, this is conducted through a very similar process for many different types of active ingredients. I will use active ingredient and AI probably interchangeably throughout my presentation today, just as a heads up if you hear me use that acronym, active ingredient, meaning AI here, not artificial intelligence. But in general, they're the same except for some specific field testing requirements, which I am going to focus a chunk of my presentation on today. Moving on to the next slide. This is something I'm certain this group is well aware of. There's many types of microbial products. These can include bacteriophages, viruses, bacteria, fungi, algae, protozoa, or perhaps interestingly, paratransgenic insect symbionts like Wolbachia. You might look at a mosquito that's been modified and not ascertain that this could be a microbial product, but EPA is regulating the active ingredient, the pesticidal substance within, in this case, the mosquito. Moving forward. We've determined what a GE microbe is and different examples of what a microbial product is and ones that could be genetically modified that would be overseen by the Emerging Technologies branch of EPA. And we also move through several regulatory statutes at our agency. I mentioned FIFRA earlier. That's one of our key statutes. It includes that definition of a pesticidal product here. And underneath of each statute here, I have the safety criteria that EPA must uphold in their risk assessment under these different statutes. So again, under FIFRA here, our safety criteria is the pesticidal product will not present an unreasonable adverse effect upon human health and the environment. It's a risk-benefit statute. Keyword there being unreasonable adverse effect. We're also under the purview of the FFDCA, Federal Food, Drug, and Cosmetic Act. This is largely under FDA's purview, but it does have some pertinence for EPA if the pesticidal product is going to be used in food or in the feed supply. The statute here has a requirement for reasonable certainty of no harm from aggregate exposure to pesticide chemical residue. And there's also PREA, which is a fee-for-service act that outlines EPA decision timeframes. This is a fee that a potential applicant would pay to the EPA, and it'll give you a timeframe for when EPA will make a decision about registering the pesticide. And also, of course, the Endangered Species Act, which has information regarding protecting threatened and listed species from environmental effects from pesticide exposure. For the next slide, I wanted to key in on FIFRA next. There's several things on this slide, several different registration paradigms. You have notifications for small-scale field testing. A lot of my presentation is going to focus here. You also have experimental use permits, special local need and emergency exemptions, pesticide registration and registration review. If you were coming into the EPA to register a commercial product, you might proceed directly to a Section 3 application, which would then be pertinent to registration review every 15 years to reassess pesticides and the risk profile at a subsequent time down the road. But in order to make a Section 3 registration, you might need additional data, additional product performance, additional testing out in the field. This could be generated either through a technology notification or an experimental use permit. I've highlighted here this notification for small scale field testing. This is what I keep alluding to being the unique thing for GE microbes under our regulatory paradigm that differentiates them from conventional pesticides. Typically for an EPA product, if you're testing on less than 10 acres of a field size, then you would not have to notify the EPA of any testing. For these microbial products, that is not the case. You would have to notify us and include some specific information before 90 days before your test would begin, if you're testing on any field size. Experimental use permits do have a large subset of regulatory data requirements that are fairly similar to Section 3 commercial registration, and those come into play if you are doing field testing on more than 10 acres of land. Next slide. The CFR goes hand in hand with FIFRA, the CFR being the Code of Federal Regulations. It's kind of like I have on the slide here, a playbook to FIFRA's statutory authority. The way we implement FIFRA is described in here, and it contains a lot of great information for how to make sure you're following FIFRA statute as an applicant as well. The Code of Regulations is quite big. 40 CFR is the one pertaining to pesticide registration. And it's part 158 subpart V, which describes the nature of microbial pesticides and those data requirements. Primarily, you'll see differences here in data requirements for microbial pesticides, GE or not, that will require additional information regarding product characterization. And again, there's special requirement for biotechnology notifications. Next slide. Getting again into our biotech notifications here. Remember again, this is 10 acres of field size or less and must be applied for 90 days before any testing begins. You can find the information that I've included on the right-hand side of the slide in the CFR under part 172, subpart C 7248. So that's a specific there. I've got the link here, which is a little less of a mouthful. But this is the kind of information you would have to supply EPA with if you're doing any field testing at all, which includes identity of the microorganism, description of the habitat, information on host range, possible transmission vectors, environmental competitiveness, description of modification methods, identity and location of the gene segments, information on the control region, description of the traits expressed, data on potential genetic transfer, description of proposed testing. Next slide. And then just two more. A statement of compositions for the formulation tested and additional information that the applicant might have on any possible adverse environmental effects. So one of the big take-home messages that I really want to provide for this specific presentation, again, is these biotech notifications, what they include, and how to find the information you need to supply the EPA with. Next slide. The microbial pesticide test requirements are described in the CFR, and you can also go to EPA's website to get these guidance documents. That webpage is also depicted here on the right side of the page. GE or non-GE microbial pesticides must comply with Series 885 pesticide guideline testing. But it's worth it to note, and quite critical for our process for GE microbes, that the EPA can ask for additional data for GE microbes depending on the unique uses of the pesticide or any unique risk concerns at any point in the process. So this would be quite important, and I will bring this message home again later. If there's another take home from my presentation today, besides information about this unique requirement for biotechnology notification, I would say the second one is communicating with the EPA often and frequently. We would highly recommend pre-submission meetings, even at the time of product concept, and especially as you're developing your data strategy. data submission strategy. So the EPA can weigh in on that and make sure we adhere to our decision timeframe by having the data we need from when the application comes in the door. The largest difference, I believe I mentioned this earlier, in terms of microbial pesticides and other conventional types of pesticides, would be that GE data requirements for microbes are subject to some specific product characterization concerns. In my laundry list of things needed to be submitted for a biotech notification, you might have heard quite a few that are pertaining to the identity of the product. One thing to note is we'll need a good taxonomic description of the microbe to compare the characteristics of closely related microorganisms to look for specific areas of concern, how closely the pesticidal product is related to known pathogens, what are the characteristics of close relatives. Gwen is going after me from the Office of Toxics, and I think she'll be talking about the structural activity relationship analysis for chemicals that go through the toxics program. This would be similar to what we might want in this regard for the taxonomic description. We want to analyze potential problems with the new chemical based on, or not new chemical here, but new pesticidal active ingredient by comparison with other known microbes. An organism that's not related to problematic microorganisms or has unique characteristics might be eligible for data waivers. Otherwise, toxins or microbes that might be known to be pathogenic to non-targets or human organisms might need additional testing to better characterize these concerns so that we can manage any possible risk or determine whether a risk exists from the microbial pesticide. Additionally, following product characterization, we would also have data requirements for human health, where surrogate species are tested for toxicity at various doses of the pesticidal active ingredient. And the same thing is done in environmental effects testing for different surrogate organisms, in this case, mammals, birds, fish, those kinds of various species. And here, I would say for environmental fate and effects, the critical part here for microbial consideration or for any product really coming through the emerging technologies branch might be keying in on environmental fate and providing good information to the Environmental Protection Agency about the persistence of your active ingredient. Lastly, I have efficacy on the slide here. I won't go into detail on efficacy here. EPA does not require efficacy data from applicants for any pesticide that is not a public health pesticide. If you're seeking a registration for a pesticide that's active against mosquitoes or another public health pest, you would have to supply efficacy data to ensure it works and to ensure public safety. But again, that's only required for these public health pests. Moving on to the next slide. I mentioned the FFDCA earlier, the Food, Drug, and Cosmetic Act. This includes information regarding rulemaking for tolerances or tolerance exemptions for food or feed uses of pesticides. Your pesticide may or may not have a food or feed use, and if so, that this statute may not apply. In general, EPA has largely seen microbial applications that have a non-toxic mode of action and generally proceed through a tolerance exemption pathway. Historically, I believe all microbial AIs have had tolerance, at least the GE ones have had tolerance exemptions. This might not be the case going forward. Of course, if in that product characterization assessment, there's any relationship with pathogenic bacteria or microorganisms or known toxins, tolerance may have to be established, but just some interesting regulatory history there might be useful for applicants. Next slide. PREA, our Pesticide Registration Improvement Act. Again, this is our fee for service category that would be applicable to GE microbes or any AI that comes through the Environmental Protection Agency. There's different PREA codes, which I have in the leftmost column, and they're associated with different actions, which could be a new AI with a tolerance exemption. You could have a new AI non-food use. You could have an experimental use permit. You can see how long it will take for EPA to communicate a decision to the applicant, and you can also see how much that will cost. It's important to note here, since I've been highlighting biotechnology notifications, that these are not a PREA action, but again, they must be submitted to the agency 90 days prior to field testing. Next slide. I want to make sure we have time for Gwen's presentation, so I'm going to move through this slide a little more quickly, but this is a review slide of everything we've talked through today, of what a pesticide application might look like. This is not specific to GE microbes or microbes in general, but pesticide applications throughout the agency. You'd have a fee for service, forms describing the requested action, product characterization, data on human health and environmental effects. This one's a big one that I'm going to discuss on the next slide as well for all AIs, but especially for products coming through the emerging technologies branch, proof that the product's manufacturing process is reliable, and of course, any labeling for pesticidal use directions. Next slide. This slide I could probably spend all day talking about, but I'll try to move through it quickly here. This is a list of best practices for registration applications that my team and the scientists on our regulatory team have observed in the last few years of emerging technologies, pesticidal applications, and things I wanted to highlight that would be good for new applicants to consider as they move through the application process. I mentioned this earlier, and I want to highlight it again here, that pre-submission meetings are highly encouraged. You can have one, you could have more than one. This could be at the time of product conception, or definitely to discuss data submission strategies so that we could discuss any case-by-case needed data, any waiver rationales, anything to that effect. These are highly useful. And at this time, you could submit meeting minutes to the agency to have us weigh in on your strategy going forward. I've mentioned biotech notifications extensively. And then these next 4 or so bullets are common issues we have with applications that you might want to have forefronted in your mind as you start making your application. Here's with the manufacturing process, making sure that this could be understood by an outside party if they were following the protocol. Of course, this is confidential business information. You won't supply these to outside parties, but filling them out as if you were training a new employee in your product development process. And then the next 3 bullets regarding meeting data requirements. Any data volume submitted to the agency must describe how the data meet the data requirements, including information about hazard, exposure, lack thereof, and citing references. And the next 2 bullets are common for microbial products. If you're submitting a literature assessment to address a data requirement in lieu of developing data or in conjunction with the developed data, it must be fully described how the publication being submitted satisfies the data requirements. And again, highlighting those things in the last 2 sentences of the previous bullet, hazard and exposure. Finally, data waiver requests. We get a fair number of these. If we do receive them, it is important to make sure they very adequately explain why the data requirement may not be appropriate for the specific new AI, why it's not possible to generate the data given the type of AI, or why it might not be useful for the agency's assessment. And there's specific guidance on our website on how to best craft these. And lastly, another issue that may be relevant for this conversation about GE microbes. If it's not possible to use 100% technical grade active ingredient, as may be the case in some of the guideline studies, it must be explained why a substitute was used or why it's not possible to generate those. And here on the last slide is a bunch of links that might be helpful to yourselves as you proceed through a regulatory process. That book chapter being a great one. That first one describes our process thoroughly. And then here are links to various guidance for data requirements or the pesticide registration process. Thank you.
As I said, we're the Office of Pollution Prevention and Toxics, and we have a biotechnology program that reviews genetically engineered microorganisms. Next slide. Okay, TSCA was actually enacted back in 1976. It was amended back in 2016 by the Frank R. Lautenberg Chemical Safety for the 21st Century Act. It gives EPA the authority to regulate the manufacture, use, distribution and commerce, and disposal of chemical substances and mixtures for chemical purposes. That also covers microorganisms that are considered mixtures of chemical substances. Next slide. We cover things that are not specifically excluded, and these are the things that are excluded from TSCA altogether. And that's products that the FDA or EPA pesticides program might have authority over. Next slide. Okay, as I mentioned, our regulation of genetically engineered microorganisms started back with the coordinated framework in 1986, which specified the lead agencies for various biotechnology products. But of course, recognizing that chemicals are very different than microorganisms, we proposed a biotechnology rule in 94, which was finalized in 97. Next slide, please. Okay, we maintained our interpretation of what a new microorganism is, as was done in the 86 coordinated framework. All of our requirements and procedures are consolidated in one part of the Code of Federal Regulations, that's 40 CFR 725. Next slide. Okay, defining what's new, TSCA has an inventory of chemical substances. If a substance is on that TSCA inventory, then it is an existing chemical. If it is not one on the TSCA inventory, it's considered new. Next slide. In addition, our concept of new microorganism is one of being intergeneric. An intergeneric microorganism is one formed by the deliberate combination of genetic material from organisms classified in different taxonomic genera. It also includes microorganisms that are constructed with synthetic genes that are not identical to the DNA that would be derived from that same genus as the recipient. And again, it's not on the TSCA inventory, and it's got to be used in a TSCA application. Next slide. Okay, we do exempt some microorganisms from TSCA reporting requirements. That includes naturally occurring microorganisms. They're implicitly listed on the TSCA inventory. We do not review intrageneric microorganisms. That means the introduced genetic materials from the same genus. And we also do not review those microorganisms that contain only well-characterized, non-coding regulatory sequences. Next slide. Okay, our definition of microorganism, according to our rule, was the Five Kingdom system of Whitaker. And this includes organisms such as bacteria, protozoa, fungi, mycoplasma, mycoplasma-like organisms, spiroplasma, microphytoplankton, green and red algae, viruses, and virus-like particles. Next slide, please. Okay, now I made some comment about TSCA uses. We have many different uses for our microorganisms. The majority of what we've seen to date has been closed system fermentation for production of enzymes, specialty and commodity chemicals. In recent years, there's been a lot for fuel production and biomass conversion, waste treatment. We do regulate biofertilizers and have been since the late 80s, bioremediation agents, biomining, oil recovery, and many, many more, including, I should mention, building materials like concrete, textiles, dyes, surfactants, all sorts of different bioproducts. And it turns out that TSCA is a catch-all statute for biotech products not regulated by other agencies. So, as I said, we have a wide variety of uses, things even such as climate modifications or all sorts of things. Next slide, please. We have two basic type of submissions we see submitted for our biotechnology products. And one is the microbial commercial activity notice, or an MCAN. That must be filed at least 90 days prior to initiating the manufacture or the import of that microorganism, unless it qualifies for an exemption. And then we have the TSCA experimental release application. And this is for persons who wish to introduce a new microorganism into the environment. And that even includes those at the R&D stage if it's deemed to be commercial research and development. And that submission has to be submitted 60 days prior to initiation of the field test. Next slide, please. I just want to run over these exemptions very quickly. We do have some research and development exemptions, but those are conducted inside a structure, a building, or if they're subject to the jurisdiction of another agency. There's also a test marketing exemption, and we also have Tier 1 or Tier 2 exemptions, which I'll cover in detail. Next slide, please. Okay, the Tier 1 and Tier 2 exemptions, it's a three-pronged approach. It's the use of an eligible recipient microorganism, and there's 4 criteria for the introduced genetic material that must be met, and there's certain containment and activation criteria that also must be met. Next slide. These are eligible recipient microorganisms for the tiered exemption. Initially, we started with 5 fungi and 5 bacteria, and these are industry workhorse microorganisms with a long history of safe use. And in 2020, we added 2 additional microorganisms to the rule, Trichoderma reesii and Bacillus amyloliquefaciens, subspecies amyloliquefaciens. Next slide, please. So these exemptions are for closed system fermentation. Here are the 4 criteria for the introduced genetic material. It must be limited in size, only to the structural genes of interest, well characterized, poorly mobilizable, and free of certain toxin sequences. There's actually a list in the rule. Next slide. With the Tier 1 exemption, aerosol releases from the fermentation facility have to be minimized, and there has to be inactivation that results in at least a 6-log reduction of viable cells in both liquid and solid waste streams. And this is a minimal notification to EPA. It's basically a postcard that says, I meet the conditions of the Tier 1 exemption. Here's the organism I'm using, and that's about it. Next slide. We have a Tier 2 exemption for those facilities that do not meet the containment requirements of the Tier 1. It could be because their inactivation is not as great, or it could be that the microorganisms are being shipped to various other facilities, like a yeast being shipped to various ethanol facilities. The Tier 1 exemption is for an individual facility only. So if you're shipping your organism, it gets kicked out of the Tier 1 exemption. That's a 45-day review, and it does require a submission to EPA to evaluate whether the proposed containment and inactivation conditions are appropriate for the microorganism. Next slide. Okay, I wanted to mention this commercial R&D. TSCA Section 5 applies to microorganisms used for commercial purposes, but we have defined commercial R&D means that the R&D activities are conducted with the purpose of obtaining an immediate or eventual commercial advantage. It can even be funded by a commercial entity, or it doesn't have to be funded by a commercial entity. But if there is an intention of making a commercial advantage, then it's considered commercial R&D. Next slide.
Hi, Gwen. You have
Yes.
about 2 minutes left.
Okay.
Yeah.
Okay, well, I will, we do have some R&D exemptions and contain structures. Next slide. Our risk assessment process is done under the paradigm that risk equals hazard time exposure. We generate these 7 reports that all get fed into one final risk assessment: the taxonomic identification report, genetic construction report, human health hazards, ecological hazards, construct hazard analysis, which looks at the genes themselves and also the potential for horizontal gene transfer, an engineering report, and an exposure assessment. Next slide. Okay, we have our guidance documents to help industry or anybody prepare their submissions for us. And this is called the points to consider in the preparation of TSCA biotechnology submissions for microorganisms. And when algae became very popular back in, around 2012, 13, 14, we ended up doing a couple workshops and developed an algae supplement that points to consider. So if someone follows these guidance documents and then also looks at the section of the rule that information to be included in either the MCAN or TERRA, then hopefully we'll get a good submission that makes it easy for us to review. Next slide. Okay, I just want to show you trend for years and years and years. We only had a few submissions and things started changing about 2013, and they're still going strong. Next slide, please. Okay, I just wanted to bring up synthetic biology. Nobody has a definition for it, but it can be simple things like a single gene change, or it can be just codon optimization. It can be metabolic engineering with replacing pathways. Next slide. Of course, there's all sorts of biologic containment strategies people are coming up with. These first two aren't necessarily synthetic biology, but then when we get into orthogonal life using xenonucleic acids or orthogonal protein translation or non-canonical amino acids, risk assessment gets a little trickier. Next slide, please. Okay, and then of course we have people who are designing minimal genome or some completely synthetic genome organisms. And when we get into that situation, we've kind of lost our comparator. We usually look at the recipient and its characteristics and then look at how the genetic modifications have changed that. But with these sort of things, we lose our comparator. Next slide. Okay, genome editing. If one of these techniques are used, unless it's a knock-in of some intergeneric genetic material, then we don't review those genetically engineered microorganisms. Next slide. Okay, this is just information, our website. And we also strongly encourage pre-notice consultations with the agency and early on in the development of your microorganism. And here are some contact information for those pre-notice consultations.